I'm Daniel Levine, and this is the Bio Report.
The emergence of tyrosine kinase inhibitors represented a major advance in the fight against cancer, but the ability of tumors to mutate and develop resistance to these therapies remains a challenge. Theseus Pharmaceuticals is developing what it calls pan-variant kinase inhibitors in the hopes of outsmarting tumors by anticipating the range of kinases that may drive their spread and growth as they change. We spoke to Tim Clackson, President and CEO of Theseus, about its structural-based approach to drug development, how it determines the appropriate targets for its pan-variant kinase inhibitors, and why it believes its next-generation TKIs can overcome the challenge of drug resistance. Tim, thanks for joining us.
It's a pleasure to be with you.
We're going to talk about resistance in cancer, tyrosine kinase inhibitors, and Theseus Pharmaceuticals' efforts to develop what it calls pan-kinase inhibitors to address treatment-resistant tumors. Let's start with kinases. What are they? How varied are they? And what role do they play in human biology?
You can think of kinases as, if you like, the master control proteins that orchestrate many important things in the cells of our body. There are about 500 kinases encoded in the human genome, and they have a huge range of jobs. But in essence, what their job is to transfer signals from one part of the cell to another part of the cell, so that the cell can respond to a signal from outside, for example. So it could be the presence of a nutrient that makes a cell grow. It could be notification that there's a different kind of cell next to that cell. So think of a kinase as a messenger that picks up an important message and takes it to where it needs to be inside the cell. They underpin essentially every kind of biology that occurs in our bodies, and so as you can imagine, they are important players.
In the case of cancers, what role do kinases play in the growth and spread of tumors?
So kinases were one of the first types of what's called oncogenes that were really fingered as being involved in cancer. So an oncogene is a gene that, when it is mutated, suddenly turns a cell into a cancer cell. The hallmark of a cancer cell is uncontrolled growth. So it doesn't know when to stop. Most other cells in our body are carefully programmed to grow when they need to grow and to stop growing when they're done. And that gets ripped up in the case of cancer. When scientists started looking for genes that could be found to be mutated in cancer, up popped kinases. So this is the normal kinase sequence that you would find in any cell, but there was a telltale mutation in that kinase. And it turns out that what that does is it basically revs up the kinase. Usually what you see is a mutation called an activating mutation that turns the kinase on and stops it turning off. So it's on all the time. And so you have, for example, a kinase that normally would tell a cell to grow. In normal physiology, it would be on for a short time, and then the cell would grow, and then the kinase would turn off and it would stop. In a cancer, it's on all the time, so the cells keep growing, and obviously bad things happen. So it turns out that many kinases are actually oncogenes, so genes that, when they're mutated, essentially cause cancer, either on their own or in combination with other oncogenes.
We've seen the advent of a class of targeted therapies known as tyrosine kinase inhibitors, or TKIs, about 20 years ago with the approval of Gleevec. How have these therapies changed cancer care, and how effective have they been?
For those tumor types where there is a very clear tyrosine kinase target, so tyrosine kinase refers to a kinase that specifically acts on an amino acid called tyrosine in proteins. If you have a situation where the disease is driven by that one kinase, TKIs have been revolutionary. So you mentioned Gleevec. Gleevec is a molecule that's been used now for over 2 decades to treat a disease called chronic myeloid leukemia, or CML. Prior to the advent of Gleevec, that's a tyrosine kinase inhibitor, those patients had debilitating chemotherapy, very, very toxic biological drugs, and unfortunately the survival was short, just a couple of years. Now, with Gleevec and subsequent molecules that have been designed as improved inhibitors of the same target as Gleevec, patients in many cases with CML can have a normal lifespan. In fact, I've had the privilege of meeting some patients who've been on that journey and have had decades of life compared to what would have been expected. So it's a revolution. And in that case where the single target of the molecule is the cause of the disease, it's been literally a lifesaver.
One challenge for TKIs has been the ability of tumors to mutate and render once effective therapies ineffective as the tumors develop resistance. What happens in those cases?
Yes, so that is the Achilles heel of any TKI, of a tyrosine kinase inhibitor, and in fact, really of any cancer treatment. And the reason is that cancers adhere to the laws of natural selection, and if you apply selective pressure that is against that cell, and in this case a kinase inhibitor that is preventing it from growing, it's going to find a way of wriggling out. The easiest way for a cancer cell that's inhibited by a TKI to become resistant is for a mutation to emerge right there in the kinase, in the target that's being bound by the drug, that stops the drug from binding. So what you invariably see in treatment, for example, with Gleevec in CML, is that you see a mutation on the target of Gleevec, and the name of that protein is BCR-ABL. And you'll see a mutation that basically stops Gleevec working. At that point, you have resistance, and there's no point in going on with Gleevec. You need a different approach. This is a pattern we've seen repeated again and again, and it can be, of course, extremely disappointing and depressing for patients who have had such a dramatic response in their initial therapy, and then to see that is subsiding.
You're taking an approach to develop what you call pan-variant targeted therapies that address all known drug-resistant mutations. What's the case for this approach?
But yes, we believe that pan-variant inhibition is the way to go. So let me take a step back and talk about what we mean by that. When you have these resistance mutations occur in a patient, they can be of many different flavors. So in treatment with Gleevec in the disease CML, scientists have documented over 50 different individual mutations that you can find on the target protein, and any one of those can stop Gleevec from working. So the approach that was taken in the years after Gleevec's development was to find better drugs that perhaps were able to overcome those mutants, those variants. And there was success in doing that, but unfortunately, we really entered into an arms race, really. It's a constant fight against the tumor. So second generation inhibitors were developed that are able to hit some of the mutations that emerge after treating with Gleevec. But then mutations emerge that create resistance to those second generation molecules. And so scientists, in fact, the group of scientists now at Theseus, in our prior company, Ariad, we developed the first third generation TKI that could overcome all of those mutations. And so that's the challenge. You need to always find a molecule that is able to hit not just one, but any of the mutations that could occur, and hence the phrase pan. So doing that, unfortunately, is easier said than done.
I think of drug developers who are working to discover new TKIs, screening large libraries of compounds against targets. What's the discovery process for Theseus? Are you scouring libraries of compounds, or are you designing molecules to hit specific targets?
Yeah, so we all of those approaches work well, and we take the approach of a large degree of what we call structure-based drug design. So that's rational design using atomic level maps of what the targets look like to understand at that high level how our drug candidates are binding. And then we tweak them, we look again at how they're binding, we test how they're working in cells, and we repeat the cycle. So I would say, instead of screening very large compound libraries and just seeing what you get, our approach is, if you like, more to handcraft the molecules and carefully engineer them to have this pan variant property. It's not something that you typically can stumble upon by just screening compounds. You have to be more structurally thoughtful.
One of the challenges of TKIs is that kinases exist throughout the body and serve important biologic functions. How much of a concern is the potential for off-target effects from such an approach?
That's always been a concern or an element of thinking for kinase inhibitors. In fact, I recall just at the time that Gleevec was developed as the first tyrosine kinase inhibitor, there was a lot of skepticism that would even be possible safely, because as I mentioned, there are 500 different kinases within humans, and there was significant concern that any kinase inhibitor would just be a sledgehammer that would have such broad effects on so many kinases in the body that it would be toxic, and you'd never have a therapeutic window, an ability to dose and get therapy. So that was proved comprehensively to be wrong, and the rest is history. It turns out that if you have an appropriately selective molecule that primarily binds to the right kinases, then you can, in fact, have great therapy with manageable side effects. However, it is generally beneficial to hit as few kinases as possible, and usually you have to think hard about which ones are most important to avoid. So this has become a key part of any kinase inhibitor discovery operation. You will carefully analyze the activity of your molecules as you're optimizing them against many, many other different kinases. Usually at least 300 of the 500 you would regularly screen against. And indeed, in many cases, you do want to spend some time to make sure you avoid targeting an undesired kinase. Having said that, we've learned a lot over the last 20 years, and we as a community of scientists and drug discoverers have now learned a lot of tricks about how to hone in just on one or a few kinases. And that's critically important if we want to reach the full potential of this molecular class, where we would aim to keep the diseases that patients have at bay for years. So chronic dosing and safety becomes very important.
Is the expectation that these therapies would be used in conjunction with some kind of tumor profiling to ensure the therapy is appropriate for a particular patient?
Tumor profiling has become a huge part of the best way to use these molecules, for sure. Another phrase that's been used to describe that is precision oncology. And this really stems from the fact that we know in many cases, CML again as another example, exactly which kinase is the bad guy. So if you can discover that mutant kinase in patients by screening, then you can know you have a much greater chance of helping that patient. Now, for a disease like CML, where the only cause of that disease is a mutation in the BCR-ABL target, you don't really need strong patient sequencing. Merely the diagnosis of CML is enough. But what's emerged over the last few years is the very exciting realization that even in more common and diffuse tumor types, so non-small cell lung cancer, for example, there are more rare patient examples where you have a very specific kinase that's been activated. And this may only be, let's say, 1% of the population. So targets known as RET or FGFR, for example, these are all kinases that are found to be activated in rare patients. If you can identify those patients, they have a much better chance of responding to those targeted drugs. And so this is the precision oncology promise. And we are thankfully at the stage now in many health centers in the US where patients coming in with a lung cancer diagnosis would be routinely screened for the presence of kinase mutations. And the hope, of course, is that becomes commonplace because it offers a great benefit and efficiency for the healthcare system to make sure you can get the right drug directly to that patient.
Well, let's talk about your lead program, THE 630, for advanced gastrointestinal stromal tumors, or GIST. What is GIST? How is it treated today? And what's the prognosis for patients with the condition?
GIST is a challenging type of tumor that's found in the gastrointestinal system, as you described. It's a sarcoma. About 5,000 patients are diagnosed with newly discovered GIST each year in the US. It's thankfully one of the types of tumors where we have a good understanding of what goes wrong. And again, it's a kinase, almost invariably a kinase that's called KIT, that gets a mutation, and that essentially causes the disease. So we've known that for 20 years. And in fact, it turns out that Gleevec, the CML drug, also has some activity against KIT. And so it's used to treat patients in the initial setting. With treatment with Gleevec, patients can proceed without any progression of their disease for at least a year and a half. So that's good, certainly much better than it was prior to TKIs. But then, just as we were discussing with other settings, you see mutations in KIT that cause resistance. And then patients are treated with a series of other molecules that don't treat the full types of mutations that you see, and the response rates are very low. Eventually, unfortunately, most patients will run out of options and succumb to their disease. So despite the fact that we know exactly what's going on in patients from a biological point of view, we've not yet come up with a molecule that's been approved yet that can finally address all of these resistance mutations in GIST.
And how predictable are the mutations once a patient's treated?
It's a pretty well-known set of mutations. So it looks as if GISTs have a limited playbook that they can follow in order to drive resistance. So there are two particular kind of initial mutations you see, and then upon treatment with Gleevec, you see another set of mutations added on top of that. But the problem is in GIST, you end up with a mixture of different mutations. So you can have one part of a patient's GIST tumor that has mutation A, if you like, and then another part close by that evolved in a different way, and that has mutation B. And in fact, in a different tumor, you might have C. So if you treat that patient with a drug that is able to hit the A and the B variants, but not the C, then you will, of course, see mutation C coming up. And it's very much like a whack-a-mole situation. So if you imagine, if you don't hit all the moles at once, then there's going to be one mole that grows rapidly. That's the problem in GIST. You have lots of mutations, lots of moles, and that's really what we've gone after with THE 630.
Well, what exactly is THE 630 and how does it work?
THE 630 is our pan variant inhibitor that we have designed carefully to be pan variant. And we've shown in our model studies that the molecule is able to inhibit any of those complex mutations that I mentioned. So the A, the B, the C that I used in the prior example. And we've shown that the molecule is able to do that at levels which we were able to safely achieve in the non-clinical setting in our preclinical models. That's in contrast to any of the currently approved agents, which includes Gleevec and 3 other approved agents. Each of those has one or more mutational Achilles heels that prevents the drug from binding. And in fact, that's what drives resistance when patients are treated with those agents. Our hope is that treatment with THE 630 will be able to break that cycle finally, and that in late stage patients where patients have already had treatment with multiple drugs, that we can rescue the disease by finally bringing all of those mutations under the control of the drug. Long term, we would have ambitions beyond that, but that's our initial hope, that we can help patients in the latest line of therapy.
Theseus began a phase 1/2 study of the THE 630 in January. This is a dose escalation study. What's known about its safety and efficacy from preclinical studies?
We were able to show in our preclinical studies that the molecule does indeed effectively inhibit each of the mutations that can be found. And that was shown both in animal models and also in careful screens that we run of cell lines, that each of which is engineered to have a specific mutation. So that's very encouraging. The other question, of course, is do we have an appropriate window for dosing the molecule safely? And we validated that in several ways, including showing that the molecule has good selectivity with respect to other molecules in the kinome, other kinases that are present in humans, and also that we were able to dose at and above the levels that we predict would give us pan inhibition. So in the clinical study, we will be looking to validate that by showing that the molecule can safely be dosed. This is an oral drug, so it's once daily pill. And we'll be looking to see whether we can sustain those pan variant levels in the blood in patients, and then to study whether we can indeed see those individual mutations being treated and going away in response to the drug. We'll hopefully have the first data from that study available in the first half of next year, in 2023.
And what's the clinical path forward?
Once we've identified the recommended dose to take forward from THE 630, that's the first aim really of the study. Then we branch out into an expansion studying the molecule in specific GIST populations. So that would include the latest line, so that's 5th line, patients who've been treated with the 4 approved therapies. We also will be interested in looking at patients in the 2nd line. So those are patients who have experienced failure of Gleevec, but have not been treated with any other agent. And the rationale for that is that if you truly have a pan-variant inhibitor, then the best place to use it is as early as possible to try and, if you like, to try and keep the genie in the bottle. You want to try and prevent mutations from coming out in the first place. That's the greatest promise of a pan-variant inhibitor, to keep mutations at bay and provide patients, we hope, with a very long time before any kind of progression can occur. So we will look at both of those populations, 5th line and 2nd line, And then after discussing with the FDA, we would design and run a registrational trial where we would hope to validate the activity and safety in those settings before applying for marketing authorization.
The company raised $100 million in a venture round in April. How far will existing funding take you?
So we are lucky enough to have funding that will allow us to clearly get to readouts from our initial clinical trial of THE 630, as well as driving other programs. In addition to our venture round earlier last year, we were able to bring in further funds through an IPO in the fall. And so together that funding takes us into the second half of 2024, which is well beyond the first half of 23 timeline, where we anticipate first data available from THE 630.
And what's the strategy for building out your pipeline? How are you going about selecting indications?
We are very attracted to settings where there's very clear biology associated with a particular tumor type, where there's no doubt about the kinase target that we need to hit, and where there's an inadequacy with current therapies. The pan-variant approach is broadly applicable to many of these settings, so the idea of finally breaking the cycle of resistance. So we've built a pipeline behind our THE 630 lead program that reflects that. We have a second program that is targeting EGFR, which is a kinase that is mutated in a substantial portion of non-small cell lung cancers. And again, we have first, second, and third generation molecules that progressively have improved treatment, but you still see resistance caused by mutations. We will be nominating our pan-variant inhibitor candidate imminently, in just a few months. And then we will conduct studies to allow us to file an IND next year, which would allow us to begin clinical testing. So our strategy is to take these molecules into the patient testing setting ourselves. And ideally, we would be looking to bring these molecules all the way through to filing for FDA approval. The Theseus team has experience of doing this in our prior work at Ariad Pharmaceuticals. And so we were looking to recapitulate that and bring the molecules to the market and to patients, certainly in the US.
And is the expectation that you'll be doing all this on your own, or are you looking to partnerships at all?
Our aim is to do it internally with respect to the US market for sure. Again, this is an approach where we have a lot of experience, a lot of understanding. We would always be open-minded as we move further down into late-stage clinical testing to collaborate potentially with partners that have expertise outside of that area. And I think that would be that's an important strategic decision that the company would take as we go further down. But our default state for sure is to seek to bring these molecules to patients, certainly in the US ourselves, and to have that as a seamless integrated process with Theseus becoming a commercial stage company in the oncology space.
Tim Clackson, President and CEO of Theseus Pharmaceuticals. Tim, thanks so much for your time today.
It's been a pleasure. Thank you.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.